In this chapter, we'll be studying the urinary system. The urinary system rids the body of waste products. The kidneys also play an important role in blood volume, blood pressure, and even blood composition. And the urinary system is closely associated with the reproductive system. In fact, they have a shared embryonic development and adult anatomical relationship and are collectively called the UG or urogenital system. In this first section, our learning objectives are to be able to name and locate the organs of the urinary system, list several functions of the kidneys in addition to urine formation, name the major nitrogenous wastes that the body creates and identify their sources, and define the word excretion and identify the systems that actually excrete wastes, as the urinary system is not the only one. Here we can see both an anterior and posterior view of the urinary system. As we can see, there are below the diaphragm here. We can also get a look at the 11th and 12th ribs and the adrenal glands, which we've already studied. And then we can see the kidneys. There's two of those, a right and a left kidney. One of the kidneys actually being a little higher than the other one because the other one has the liver on top of it with a little bit of a lower lobe that it has to make room for. We can see the tubes coming from the kidneys. Those are the ureters and the urinary bladder and the urethra. Also associated, we can see what's called the renal artery and the renal vein, which bring blood in and out of the kidneys. When you hear the word renal, think kidney. And also for perspective, we see the aorta and the inferior vena cava as well. There's actually quite a few functions of the kidneys. It's not just all about getting rid of wastes, but indeed that will be one of the biggest areas that we focus on in this chapter for function number 1, filtering blood plasma and excreting toxic wastes. The kidneys also regulate blood volume, pressure, and osmolarity. Remember, osmolarity has to do with the amount of solutes in the solutions of the body. They help to regulate electrolytes and acid-base balance. They secrete erythropoietin, which, as we already know, stimulates the production of red blood cells. They help to regulate calcium levels by participating in calcitriol synthesis. They clear hormones from the blood, detoxify free radicals, and in starvation, they can synthesize glucose from amino acids. The synthesizing of glucose from amino acids is called gluconeogenesis, and not many organ systems can do this. It's actually quite expensive energy-wise to do it, but remember that brain needs glucose. It can't use any other fuel. So in times of starvation or in severe glucose depletion, your kidneys actually can take products like amino acid and turn them into glucose, which might be very much needed by the brain. This chart is not from your book. These are some numbers I put together. Kind of some food for thought. We'll start over here with the water out. So this is the amount of water that comes out from your body in a typical day. Keep in mind this is just normal, not doing anything too strenuous type of activity day. No extra exercise or anything like that. So these numbers would obviously change in the event of doing exercise or certain labor. So 1.5 liters of urine is excreted every day. This is the bare minimum. This is the minimum amount needed so that you can remove metabolic waste from the body. You have to excrete 1.5 liters of urine a day to be healthy and to get rid of the metabolic waste. That is the bare minimum right there, 1.5 liters. 100 milliliters is lost usually in a day in sweat. Again, that's not exercising or anything like that. 200 milliliters is in the feces. 350 milliliters through the skin. The skin isn't a complete barrier. There are pores and things do go through the skin, including water. And 350 milliliters is lost in the lungs when you breathe out. Like if you breathe on a window or a mirror, it leaves the water drops that's coming from your body. And in a day, if you could collect that all and add it all up, it would be about 350 milliliters. So that's the bare minimum. This is what I call the daily obligatory water loss. These are all minimum numbers once again. So in order to make up for that water just normally lost in a day, you need to compensate that with at least 2.5 liters. Also, it converts to 2,500 milliliters if that helps. So keep those numbers in mind as we go through the chapter. The kidneys and urinary system is known as a retroperitoneal system. This is when you're outside of the peritoneum. You follow the peritoneum right here. You can see the pancreas is also retroperitoneal on the outside here. Here's the kidneys. We can see the vertebrae there. So if you were to say do a surgery and you enter on the anterior side and you got all the way back here, the kidneys, including the pancreas as well, all of this would be covered by peritoneum. You would have to enter the peritoneal cavity or go through the peritoneal cavity to the outside of it to get to the kidneys there. A waste is any substance that's useless to the body or present in excess. And a metabolic waste is a substance that's produced by the body. Your cells are doing their normal daily work, and the metabolic wastes are what they produce as they do their normal daily work. And these are the main metabolic wastes that your kidneys need to remove. Urea is the big one, including ammonia there. Proteins and amino acids have NH2, which is then removed and then forms ammonia. Here we can see the ammonia molecule there, and you can actually see it in two places in the urea molecule. Those two are the big ones we'll look at in the chapter. The ammonia is so extremely toxic, your cells make it, you have to get rid of it. Your liver will take ammonia and then turn it to urea. Now, uric acid is a product of nucleic acid catabolism. Here's the molecule right there. Birds and reptiles, interestingly enough, convert urea to uric acid. It's metabolically expensive to do this, but the benefit is that then it doesn't take much water to get rid of uric acid, so their urine can be much more concentrated, and they don't have to carry around so much excess water while they're flying. So if you've ever seen bird poop or reptile poop, they combine their uric acid with the feces. Then there's creatinine. That's a product of creatine phosphate catabolism, molecule right there. You don't have to memorize the formulas of these molecules or anything, but do know that these are the main nitrogenous wastes that our body, especially the kidneys, must remove. If they were to build up, they would become toxic. Blood urea nitrogen, also known as BUN, is the level of nitrogenous waste in the blood and can be measured. Its normal concentration should be about 10 to 20 milligrams per deciliter. If you have azotemia, you have elevated BUN and can indicate that your kidneys aren't removing enough metabolic waste, and so you have renal insufficiency. Uremia is a syndrome of diarrhea, vomiting, dyspnea, cardiac arrhythmia that stems from the toxicity of nitrogenous wastes. The treatment needed for this would be hemodialysis. We'll talk about that at the end of the PowerPoint. And also organ transplant might be needed as well if those kidneys are not functioning like they should. Just keep in mind any of these that build up, they become toxic level and can lead to kidney shutdown and death. Excretion is separating the waste from the body's fluids and then eliminating them. And you have a number of body systems that carry out excretion. We've already studied the respiratory system, which removes carbon dioxide, small amounts of other gases, and water. You've already studied the integumentary system in Anatomy and Physiology 1, which can remove water, inorganic salts, lactic acid, and urea within sweat. We've already studied the digestive system, which removes water, salts, carbon dioxide, lipids, bile pigments, cholesterol, and other metabolic wastes. And finally, the urinary system, which can get rid of many metabolic wastes, toxins, drugs, hormones, anything that you might come across that enters your body that's unrecognized or unneeded, other hormones, salts, even hydrogen ions, and water. The removal of those hydrogen ions helps us with our pH balance. In this section, our learning outcomes are to describe the location and general appearance of the kidney, identify the external and internal features of the kidney, trace the flow of blood through the kidney, trace the flow of fluid through the renal tubules, and describe the nerve supply to the kidney. The kidneys lie against the posterior abdominal wall at the level of the T12 and L3 vertebrae. The right kidney slightly lower due to that large right lobe of the liver. Rib 12 actually crosses the middle of the left kidney. And again, they're retroperitoneal, along with the ureters, the urinary bladder, the renal artery and vein, and the adrenal glands. They're about the size of a bar of bath soap, and the lateral surface is convex, and medial is concave with a slit called the hilum. It receives renal nerves, blood vessels, lymphatics, and the ureter. There's 3 protective connective tissue coverings. The renal fascia is immediately deep to the peritoneal or parietal peritoneum, and it binds the urinary system to the abdominal wall. The pararenal fat capsule cushions the kidney and holds it in place, and there's also a fibrous capsule that encloses the kidney itself, protecting it from trauma and infection. There's collagen fibers that extend from the fibrous capsule all the way to the renal fascia. And still, even when you lie down and then stand up, there's still about a 3 centimeter drop when changing those positions. So there is still even a little bit of flexibility in there. The renal parenchyma is the glandular tissue that forms urine. And we'll talk about the different structures within the renal parenchyma. It appears C-shaped in a frontal section and encircles the renal sinus. The renal sinus is the cavity that contains blood and lymphatic vessels, nerves, and urine collecting structures. Adipose fills the remaining cavity and holds the structures in place. There's two zones of renal parenchyma, the outer zone, which is called the renal cortex, and the inner zone is called the renal medulla. The renal medulla has what are called renal columns and renal pyramids. The columns are extensions of the cortex that project inward toward the sinus, and the pyramids have 6 to 10, and there's a broad base facing the cortex and the renal papilla facing the sinus. The lobe of the kidney is one pyramid and its overlying cortex. Other structures include the minor calyx, which it's a cup that nestles the papilla of each pyramid and collects its urine. So it's kind of a collecting area. And then the major calyces are formed by the convergence of 2 or 3 minor calyces. The renal pelvis is formed by the convergence of 2 or 3 major calyces, and the ureter is a tubular continuation of the pelvis that drains urine down to the urinary bladder. In this diagram, you can locate the structures we've just mentioned, including the fibrous capsule, the cortex, which is the outer layer, the medulla, the inner layer, the renal papilla, the renal sinus here, the renal pelvis, the major calyx. See how the pelvis is a collection of a number of major calyxes. Here's a minor calyx. Here's the renal column, a renal pyramid here, and in the cross section we can see some renal blood vessels as well. And here's the ureter. Kidneys are only about 0.4% of the body weight, but being such small structures, they receive about 21% of the cardiac output of blood from the heart. That's called the renal fraction. So at any given time, over 20% of your blood is in your kidneys. Blood enters the kidney via the renal artery, and that's going to divide into smaller and smaller segmental arteries until finally they branch into what's called an afferent arteriole. Each of those is going to supply one nephron and will lead to a ball of capillaries called the glomerulus. This is going to be where the major filtration happens, pulling out waste and other materials from the blood. But we'll get to that more in our next section. Blood then will be drained out of the glomerulus by the efferent arteriole, and that'll branch into bigger and bigger arterioles. It'll lead to the peritubular capillaries, and we'll see how the peritubular capillaries surround the nephron tubule. A nephron is a filtering unit of a kidney. So we'll see how these blood vessels, these arterioles, capillaries, and venules will kind of branch around these filtering units, and this is going to allow the nephron to pull items out of the blood and vice versa, be able to put things back into the blood. We have to keep a very close connection or a close association with the nephron and the blood vessels. Some of the efferents will lead to what's called the vasa recta, and that's a network of blood vessels within the renal medulla, so they're just a little bit lower in the kidney. And then the capillaries will eventually branch into larger veins and eventually leave the kidney via the renal vein, which will then empty into the inferior vena cava. So after blood goes through the kidneys, it actually goes right back to the heart. Here you can follow that circulation all the way from the heart and the aorta I left out some of these other intermediate ones in here. You don't have to know those, but you should know that blood comes in through the renal artery, branches to segmental arteries, then down to the afferent arteriole, which leads into the glomerulus. Make sure you know the efferent arteriole and the peritubular capillaries. I left these three out here. Do know that it exits the renal vein and then goes to the inferior vena cava. We also did mention the vasa recta as well. Here we can actually see some pictures of nephrons. Anything in yellow here, that's the nephron, these curly, windy structures. And then you can see the close association with the blood vessels, still showing arteries and arterioles in red, and capillaries here, the smaller ones, venules and veins blue. Up here, in this area here, this is the cortex. Down in this area, it's the medulla of the kidney. So you can see some of these nephrons curl and wind way low down into the medulla, while some only just a little bit wind down into the medulla. The ones that, these nephrons that wind all the way down into the medulla, very low, have a long, what's called nephron loop, are called juxtamedullary nephrons, while the ones that don't go as low are called cortical nephrons. Then you can see how they wind and eventually empty into this collecting duct here. And one collecting duct might serve a number of different nephrons. In fact, this collecting duct serves a juxtamedullary nephron here and a cortical nephron there. When something comes from the nephron and then gets emptied into the collecting duct, it's then called urine. In the cortex here, we can see the peritubular capillaries that branch off from the efferent arterioles that supply the tissue near the glomerulus, and the proximal and distal convoluted tubules. So those are signified with PCT, proximal convoluted tubule, and distal convoluted tubule, PCT and DCT for short. These capillaries that continue and have a very close association with these tubules are called the peritubule capillaries because they're around the perimeter of all these tubules here. In the medulla, the efferent arterioles give rise to the vasa recta, so that's here. So when these tubules go way down deep into the medulla like this, then they're called the vasa recta, and they supply the nephron loop portion of the nephron. Each kidney has about 1.2 million nephrons. Again, each nephron, think of that back here, each one of these is a filtering unit. Each kidney has 1.2 million on average. And the nephron has two principal parts. There's the renal corpuscle. This is what filters the blood plasma. And the renal tubule, which is that, like we already talked about, the long tubes here, some a little bit longer than others, that even wind up and around. And again, here's that glomerulus and the capsule around it. That whole area is the renal corpuscle. So the renal corpuscle consists of the glomerulus and a two-layered glomerular capsule that encloses the glomerulus. The parietal is the outer layer. It has simple squamous epithelium. And the visceral is the inner layer and has what are called elaborate cells called podocytes that wrap around the capillaries of the glomerulus. And the capsular space separates the two layers of the glomerular capsule. So here's our renal corpuscle. Our afferent arteriole brings blood in. It's under a great amount of pressure once it's in this capillary bed called the glomerulus inside here. And so filtration happens quite easily here. The filtrate then occupies this capsule space and then exits via the proximal convoluted tubule. Oh, we're also supposed to note how the afferent arteriole is larger than the efferent arteriole. That's going to play a role a little bit later. The vascular pole is the side of the corpuscle where the afferent arteriole enters and the efferent leaves it. So the pole is here. And the urinary pole is on the opposite side of the corpuscle where the renal tubule begins over here. The renal tubule is the duct leading away from the glomerular capsule and then ends at the tip of the medullary pyramid. It's divided into 4 regions. So this whole network of winding tubes starts with the proximal convoluted tubule, or PCT for short, the nephron loop. This used to be called the loop of Henle. Then the distal convoluted tubule, or DCT for short. Those are all parts of one nephron. And then there's the collecting duct. Like we've seen in the picture, one collecting duct can receive from many nephrons. And I'll jump back to this picture here. So leaving this nephron here into the proximal convoluted tubule, then the nephron loop. If I call it the loop of Henle, you'll know it's the same thing. That's what they called it in my day, but they changed that name. They just removed someone's name out of there. Actually makes it a little bit easier. And then the distal convoluted tubule here, and then the collecting duct, which serves not only this juxtamedullary nephron, but this cortical nephron as well. The proximal convoluted tubule arises from the glomerular capsule, and it's the longest and most coiled region. It has simple cuboidal epithelium with prominent microvilli for absorption. The nephron loop is the long U-shaped portion of the renal tubule. It has a descending limb and an ascending limb. And then it has I'll go back here. Here's the loop, and here's the descending, and then the ascending. And it has a thick area on each side, and then in this bottom portion it has a thin area. The thick segment has simple cuboidal epithelium, and its cells, because it's got to do a lot of active transport and that requires ATP, they have a lot of mitochondria where you've got much cell respiration going on, creating ATP. The thin segment has squamous epithelium, and it's that lower portion of the descending limb. Here the cells are very permeable to water. In the distal convoluted tubule, it begins shortly after the ascending limb re-enters the cortex. It's shorter and a little bit less coiled, has cuboidal epithelium with no microvilli, and is considered the end of the nephron before it empties into the collecting duct. The collecting duct receives that fluid from the DCT of several nephrons as it passes back into the medulla. So if we were to follow the flow from one point where the filtrate is in the glomerulus, where it's formed, to where the urine leaves the body, it would be this whole collection in this order, from the glomerular capsule to the proximal convoluted tubule, down through the nephron loop, the thick, thin, and thick again, ascending to the distal convoluted tubule, here to the collecting duct. From there, the papillary duct to the minor calyx, to the major calyx, the renal pelvis, then the ureter, and then the urinary bladder, and then finally the urethra, and then eventually out of the body. Here's that same picture blown up a little bit bigger with everything labeled for us. So you should be able to trace the path of that filtrate out of the body. Again, this is a pretty simplified version. It might look a little bit more like this if we were looking at it in a real kidney. There's, with the cortical nephrons, they make up about 85% of all nephrons with short loops, and the efferent arterioles branch into peritubular capillaries around the PCT and DCT. Remember, the juxtamedullary nephrons are the ones with the long nephron loops that go down into the medulla. That's 15% of all nephrons. Those long nephron loops maintain a salinity gradient in the medulla and help us conserve water. And the efferent arterioles branch into the vasa recta instead of the peritubular capillaries. The renal plexus is what innervates kidneys. It's the nerves and the ganglia wrapped around each renal artery. It follows the branches of the renal artery into the parenchyma of the kidney and issues nerve fibers to blood vessels and convoluted tubules of the nephron. It carries a sympathetic innervation from the abdominal aortic plexus, and its stimulation reduces glomerular blood flow and then therefore the rate of urine production. It can respond to falling or low blood pressure by stimulating the kidneys to secrete renin, which is an enzyme that activates hormonal mechanisms to restore blood pressure. In this section, we're going to look at urine formation. Part 1 mainly in the glomerulus and filtration there. In our next section, we'll look at another portion of that within the tubule. Our expected learning outcomes are to describe the process by which the kidney filters the blood plasma, including the relevant cellular structure of the glomerulus, to explain the forces that promote and oppose filtration, and calculate the filtration pressure if given the magnitude of those forces, and to describe how the nervous system, hormones, and the nephron itself are able to regulate filtration. Here's the basic stages of urine formation. Number 1, in here in the renal corpuscle, we can find, this is the capsule, we can find the glomerulus in there. It filters our blood and creates a plasma-like filtrate. Then it goes into the proximal convoluted tubule. Here that tubule removes useful solutes from the filtrate and then returns them to the blood. As the filtrate moves more through the tubule, secretion will occur. That's using active transport. It'll remove additional waste from the blood and then add them to the filtrate. Then water conservation. This is our body's way of keeping the amount of water balanced. So either here we're going to conserve water by putting it back into the blood, or we're going to leave it in the tubule and send it to the collecting duct and urinate it out. So this is also called concentrating wastes. if you are pretty dehydrated, you'll notice your urine is also quite concentrated. That's because in this last portion here, you're actually conserving water and sending it back to the blood as much as possible. Whereas if you've had enough to drink during the day, you've drank extra, maybe you've been drinking coffee all morning or something like that, you'll actually be able to afford to get rid of more water, and so your urine will be less concentrated. It'll be more dilute. So the kidneys convert blood plasma to urine in 4 stages: glomerular filtration, tubular reabsorption, secretion, and then water conservation. The glomerular filtrate is the fluid that's in the capsular space. It's similar to blood plasma, but it doesn't have the amount of proteins. Proteins are big molecules. They tend to stay in the blood and not get filtered out, as well as the blood cells are too big. They're not going to be filtered out either. The tubular fluid is the fluid from the proximal convoluted tubule all the way through the distal convoluted tubule. And then the substances there have been either removed or added by tubular cells. And then urine is the fluid that's in the collecting duct. Anytime filtrate enters that collecting duct, it will become urine. Here's a neat picture. So if this is the blood vessel here, we can see the blood vessel is fenestrated. It has holes in it. So it's not just about diffusing this stuff across the cell membrane, also going through these holes as well. So what's not going to get out are blood cells, plasma proteins like albumin and such, large anions, protein-bound minerals. So those are minerals or other substances that attach themselves to the proteins. And then anything that's 8 nanometers in diameter or more can't get through these little pores, can't go across the cell membrane, so they stay in the blood. But items like water easily gets through, electrolytes, glucose, amino acids, fatty acids, vitamins, urea, uric acid, and creatine can get across and get into the capsular space. The filtration membrane has 3 barriers through which the fluid has to pass. It has to go through those fenestrated glomerular capillaries. Again, they're highly permeable to water and such, but not to proteins or blood cells. The basement membrane, which is the proteoglycan gel. Proteoglycan is kind of a network of glycoproteins hooked onto some core proteins. And then all of that has a negative charge, which helps to repel molecules greater than the 8 nanometers. Albumin, the protein, is also repelled by that negative charge. So that stays in the blood. Blood plasma is about 7% protein, so as a comparison, the filtrate that enters the capsule then, capsular space, is only about 0.03% protein, so only some small ones are going to get through there unless there's other problems that the kidney has, which we'll talk about. There's also filtration slits, which have the podocyte cell extensions that wrap around the capillaries to form the barrier layer with 30 nanometer filtration slits. It's negatively charged, which again, that's one of those obstacles for anything that's like a large anion or a negatively charged particle that's in the blood. Glomerular filtration is a special case of capillary fluid exchange in which water and some solutes in the blood plasma pass from the capillaries of the glomerulus into the capsular space of the nephron. So almost any molecule smaller than 3 nanometers could get through, and some substances of low molecular weight are bound to the plasma proteins and can't get through, and so that actually keeps calcium, iron, or iron and thyroid hormone in the blood. Kidney infections and trauma can damage that filtration membrane, and this is when other things are allowed into the filtrate that shouldn't be, like blood cells, and albumin. So proteinuria is presence of protein in the urine, like albumin. And hematuria is the presence of blood in the urine. Unfortunately, distant runners and swimmers often experience these two disorders because of such strenuous exercise, reducing the perfusion of the kidney, and the glomerulus deteriorates under the prolonged hypoxia. Usually it's just temporary, and they will heal. Filtration pressure depends on hydrostatic and osmotic pressures on each side of the filtration membrane. Glomerular filtration follows the same principles that govern filtration in other blood capillaries, but there's some differences in the magnitude of forces. The blood hydrostatic pressure, or BHP, is much higher here than elsewhere. It's about 60 millimeters of mercury compared with 10 to 15 millimeters of mercury in most other capillaries of the body. And then this results from the fact that the afferent arteriole is substantially larger than the efferent arteriole. It gives the glomerulus a large inlet but a small outlet. The hydrostatic pressure in the capsular space is about 18 millimeters of mercury, compared with the slightly negative interstitial pressure elsewhere. This results from the high rate of filtration and continual accumulation of fluid in the capsule. The colloid osmotic pressure, or COP for short, of the blood is about the same here as anywhere else, about 32 millimeters of mercury. The glomerular filtrate is almost protein-free, and so therefore doesn't have a significant COP. But it could change markedly in kidney diseases that allow proteins into the capsular space. So then on balance, we have a high outward pressure of 60 millimeters of mercury, opposed by two inward pressures of 18 and 32 millimeters of mercury, which gives a net filtration pressure, or NFP, of 60 out minus 18 in minus 13 in of 10 millimeters of mercury out, shown in the diagram like this. High blood pressure in the glomerulus makes kidneys very vulnerable to hypertension and can lead to rupture of the glomerular capillaries, which could produce scarring or nephrosclerosis. This also promotes atherosclerosis of the renal blood vessels and could ultimately lead to renal failure. So again, think of that large afferent arteriole bringing that blood in here. It's bigger, so you have that greater pressure versus the smaller one with the blood trying to leave here. So all that pressure causes more to go out into the capsule space than what's coming in. So again, NFP out is 10, which we will need for this equation. Glomerular filtration rate, or GFR, is the amount of filtrate formed per minute by two kidneys combined. For every 1 millimeter of mercury of net filtration pressure, the kidneys of a young adult male produce about 12.5 milliliters of filtrate per minute. So this value is called the filtration coefficient, or KF for short. That'll be this number here. And it depends on the permeability and the surface area of the filtration barrier. KF is about 10% lower in women than in men. So then we have our equation here, NFP times the filtration coefficient For men, that's 125 milliliters per minute, and it'd be 180 liters per day. That is a big number. For females, it's 150 liters per day. The total amount of filtrate produced per day equals 50 to 60 times the amount of blood in the body. So 99% of that filtrate is reabsorbed. So think of that. If you were to lose, if you're a male, you're losing 180 liters a day from your bloodstream. Females 150 liters a day. Could your body withstand that? Absolutely not. That's more blood than you have in your body. So literally, even though so much fluid is pulled out of the blood daily, you actually put most of that fluid back. 99% of that filtrate is reabsorbed. So when we say reabsorbed, it means it's sent back to the blood. So yes, it's pulled out in the glomerulus, but as that filtrate moves along the tubule in close association with the blood vessels, it'll be reabsorbed or put back to the blood. So literally only about 1 to 2 liters of urine will be excreted per day. If GFR is too high, then fluid flows through the renal tubules too rapidly for them to reabsorb the usual amount of water and solutes, so the urine output rises. But then there's a chance of dehydration and electrolyte depletion. If it's too low, then wastes are reabsorbed and azotemia might occur. So GFR is controlled by adjusting the glomerular blood pressure from moment to moment. GFR control is achieved by 3 homeostatic mechanisms: renal autoregulation, sympathetic control, and hormonal control. The nephrons themselves can adjust their own blood flow and GFR without any other external control, meaning without control from the nervous or hormonal systems. That's called renal autoregulation. It enables the kidney to maintain a relatively stable GFR in spite of changes in systemic blood pressure. There's 2 methods of autoregulation: myogenic mechanism and tubuloglomerular feedback. The myogenic mechanism is all about stabilizing the GFR. It's based on the tendency of smooth muscle to contract when it's stretched. So if the arterial blood pressure increases, the afferent arteriole is stretched, and then the afferent arteriole constricts and prevents blood flow into the glomerulus from changing. So it helps again to stabilize it. If the arterial blood pressure falls, then the afferent arteriole relaxes, then the afferent arteriole dilates and allows blood flow to go into the glomerulus more easily, so that the flow rate remains similar and the filtration again remains stable. The tubuloglomerular feedback is when the glomerulus receives feedback on the status of downstream tubular fluid and adjusts filtration rate accordingly. It regulates the filtrate composition, stabilizes kidney performance, and compensates for fluctuations in blood pressure. And it has to do with this area right in here called the juxtaglomerular apparatus. So what you're looking at here, here's the glomerulus. Here's the efferent and the afferent arteriole. And here, the nephron loop is cut into a cross section. See how that nephron loop is winding its way back up and comes in such close quarters with the efferent and the afferent arteriole. So the juxtaglomerular apparatus is a complex structure found at the end of the nephron loop, where it just has re-entered the renal cortex, and it comes into contact, such close association with the efferent and efferent arterioles, at the vascular pole of the renal corpuscle. So the macula densa is a patch of slender, closely spaced sensory cells in the nephron loop. We can see those cells right here in the nephron loop. When GFR is high, the filtrate contains more sodium chloride. So then the macula densa there absorbs more sodium chloride, and then it secretes ATP. The ATP is metabolized by nearby mesangial cells into adenosine, and then adenosine stimulates nearby granular cells. Here's the mesangial cells, and here's the granular cells. The granular cells are actually modified smooth muscle cells wrapping around the arterioles, and they can respond to adenosine by constricting the afferent arteriole. So then that constriction would reduce blood flow, which would then correct the GFR. The mesangial cells might also contract, constricting capillaries, which further limits the GFR. Those granular cells also contain granules of renin, which they can secrete in response to a drop in blood pressure, and they can participate in the renin-angiotensin-aldosterone system that works to control blood volume and pressure. Renal autoregulation regulates GFR, but it can't keep it entirely constant. Rises in blood pressure will cause a rise in GFR, but if mean arterial pressure drops below 70 millimeters of mercury, then filtration and urine output will cease. For instance, in like an elderly individual who is dying, if their systolic blood pressure gets too low, then there's not enough pressure for filtration to occur, and then that's the beginning of renal failure, and they're not pulling out of their blood anymore all of those metabolic wastes. If the GFR rises, more NaCl is reabsorbed, and more adenosine is produced locally. The adenosine stimulates the JG cells to contract, which constricts the afferent arteriole, which reduces GFR to normal. If GFR falls, the macula relaxes the afferent arterioles and the mesangial cells, and blood flow increases and GFR rises back to normal. Here we can follow the negative feedback control of the GFR. High GFR leads to increased sodium chloride in the nephron loop. The macula densa releases ATP. The mesangial cells metabolize ATP to adenosine. This stimulates the granular cells, which then causes the afferent arteriole to constrict and reduce GFR. Sympathetic nerve fibers richly innervate the renal blood vessels. Sympathetic nervous system and adrenal epinephrine can constrict the afferent arterioles in strenuous exercise or in acute conditions like circulatory shock, which reduces GFR and urine output. It redirects blood from the kidneys to the heart, the brain, and the skeletal muscles, because in those times, making urine is not as important as getting blood to the heart, brain, and skeletal muscles. GFR could fall quite low in times of exercise or circulatory shock. The renin-angiotensin and aldosterone mechanism. It's a system of hormones that helps to control blood pressure and GFR. Got those baroreceptors again in the carotid and aorta sinus. Stimulates the sympathetic nervous system when a decrease in blood pressure is detected. Then the sympathetic fibers trigger the release of renin by the kidneys in those granular cells. And then renin converts angiotensinogen, which is a blood protein, into angiotensin 1. In the lungs and the kidneys, there's an enzyme that converts the angiotensin 1 into angiotensin 2, which angiotensin 2 is the active hormone that increases BP by doing quite a few different jobs here. The simplest is on the bottom, just stimulating thirst. So if you drink more, you'll add more water to your blood and then increase blood pressure that way. But other things that it does, it's a potent vasoconstrictor, which raises BP. It constricts the efferent arteriole, which raises the GFR. It lowers the blood pressure in the peritubular capillaries and enhances reabsorption of sodium chloride in water. We bring more water back into the blood from the kidney, then our blood pressure should go up. It stimulates the adrenal cortex to secrete aldosterone, which promotes sodium and water reabsorption in the distal convoluted tubule in the collecting duct. Again, we're keeping more water around in our blood instead of urinating it out, so this should help bring up blood pressure. It stimulates sodium and water reabsorption in the proximal convoluted tubule, and it also stimulates the posterior pituitary to release the antidiuretic hormone, or ADH, which promotes water reabsorption by the collecting duct as well. All of those bringing up blood pressure. In this section, we're going to look at urine formation again, but concentrate more in the tubular regions on reabsorption and secretion. Our outcomes are to describe how the renal tubules reabsorb useful solutes from the glomerular filtrate and then return them to the blood. To describe how the tubules secrete solute from the blood into the tubular fluid, and to describe how the nephron regulates water excretion. Conversion of glomerular filtrate to urine involves the removal and the addition of chemicals by tubular reabsorption and secretion, and this is going to occur all the way through the proximal convoluted tubule, through the nephron loop, all the way to the distal convoluted tubule. And throughout this whole region, the tubular fluid is modified in these steps: tubular reabsorption, secretion, and water conservation. The proximal convoluted tubule reabsorbs about 65% of the glomerular filtrate. Remember, by the end of this, we want to bring 99% back into the blood. But here in the beginning of the tubule, we'll get about 65% of it back. It removes some substances from the blood and secretes some into the tubular fluid for disposal in urine. Again, this area has a lot of mitochondria to do active transport to make ATP. And so this area, because of all the active transport going on, this area makes up about 6% of your resting ATP and calorie consumption. So tubular reabsorption is the process of reclaiming water and solutes from the tubular fluid and putting them back into the blood. Again, we couldn't afford to lose all the fluids and items that are filtered out initially. We have to put them back without putting the waste back as well. We want to get rid of the waste part. There's two routes of reabsorption: transcellular, meaning the substances can pass through cytoplasm of the epithelial cells and then out of their base. So transcellular is going through the cells. Paracellular is going between those epithelial cells. Luckily, those junctions there are pretty leaky and allow things to go through quite well, especially water. That creates solvent drag because water carries a variety of dissolved solutes with it as well. Reabsorbed fluid is ultimately taken up by the peritubular capillaries. Sodium reabsorption is a big key to all of this. Sodium creates an osmotic and electrical gradient that drives the reabsorption of water and other solutes. Water likes to move from where there's more water to less. It likes to dilute out solutes. So water will often follow sodium in order to dilute it out. This creates a steep concentration gradient that favors its diffusion into epithelial cells. There's two types of transport proteins in the apical cell surfaces that are responsible for the sodium uptake. Symports, those are protein carriers that simultaneously bind sodium and another solute, such as glucose or amino acid. So symport means that it's pulling two items across together. Sodium and hydrogen antiport, that means it's a switch, bringing sodium into the blood in exchange for putting hydrogen from the blood to the tubule, is antiport. And this is going to also be key in getting rid of hydrogen ions from our body and helping to maintain pH. So again, it's this important sodium pump. That's important because if we pump sodium back into the blood, water will follow. And vice versa, if we pump sodium into the tubule, water will follow that way. Sodium is picked up by the peritubular capillaries and returned to the blood. And the sodium-potassium pump at the bases are examples of primary active transport because they're using ATP, but the symports on the apical surface are examples of secondary active transport. They're not really using ATP, but are dependent on those primary transport of sodium and potassium pumps at the base of the cell to establish that sodium concentration gradient. The negative chloride ions follow the positive sodium ions just by electrical attraction, opposites attract. And various antiports in the apical cell membrane that absorb chloride in exchange for other anions that they eject into the tubular fluid are called the potassium chloride symport. Potassium, magnesium, phosphate ions diffuse through the pericellular route with water. Phosphate is also co-transported into the epithelial cells with sodium, and some calcium is reabsorbed through the pericellular route in the proximal convoluted tubule, but most calcium is reabsorbed, occurs later in the nephron. Glucose is co-transported with sodium by SGLT proteins, sodium glucose transport proteins. And normally all the glucose that's taken out of the blood is reabsorbed or put back. Unless you're diabetic, then you have too much glucose for all the transporters. They can't keep up, so ultimately glucose is or a bunch of glucose is going to stay in the urine and it'll be excreted out. That testing the glucose in the urine is one of those tests for diabetes. For the nitrogenous wastes, the nephron reabsorbs about half of the urea in the tubular fluid, but that's okay because that concentration is safe for the blood. And the proximal convoluted tubule reabsorbs uric acid, but later portions of the nephron will secrete it, meaning they'll grab it back out again and get rid of it. Creatine is still passed in the urine. It's not reabsorbed at all. Each day, kidneys produce about 180 liters of glomerular filtrate to 1 to 2 liters of urine. It's amazing. 2/3 of the water in the filtrate is reabsorbed in the proximal convoluted tubule. So 2/3 of the water pulled out at the glomerulus is put back in the PCT. Reabsorption of solutes makes the tubule cells and the tissue fluid hypertonic to the tubular fluid. And water follows solutes by osmosis through both pericellular and transcellular routes. So transcellularly, the water will go across using protein channels called aquaporins, very appropriately named. So in the PCT, water is reabsorbed at a constant rate called the obligatory water reabsorption. It goes back to that number I gave you earlier. It's that certain amount of water that you must excrete every day to be able to mix with the waste to get rid of those wastes from the body. There's just no getting around that. So what they're saying here is by putting solutes back into the blood, it makes water follow those solutes by osmosis. Water will move from where there's more water to less water. That's the idea of osmosis across cell membranes. And so if we pump solutes back into the blood, water will follow that. And then the water itself will also end up back in the blood. The peritubular capillaries reabsorb water and solutes that leave the basal surface of the tubular epithelium. And reabsorption here occurs by osmosis and solvent drag. Three factors that promote osmosis into the capillaries: a high interstitial fluid pressure due to the accumulation of reabsorbed fluid in the extracellular space, makes water want to go back to the blood. Low blood hydrostatic pressure in the peritubular capillaries due to the narrowness of the efferent arteriole. Remember, water would like to go from high pressure to low, considering its own self. So it'll go from where it's high in the interstitial fluid in the tubule to where it's low in the blood. And then high colloid osmotic pressure in the blood with the proteins there. Remember, those proteins were not filtered out in the glomerulus. So again, high osmotic pressure there. That also draws water back in. The amount of solute that renal tubules can reabsorb is limited by the number of transport proteins in the tubule cells of the membranes. If all transporters are occupied, any excess solute passes by and then appears in the urine. The transport maximum is reached when the transporters are saturated, and each solute has its own transport maximum. So any blood glucose, for instance, level above 220 milligrams per deciliter will result in glycosuria, which is glucose in the urine. So people who have, for instance, type 2 diabetes, have a higher blood sugar. You don't have enough glucose transporters to put that back into the blood as the filtrate is going by. So some of that glucose remains in the tubule, ends up in the collecting duct, and then ends up in the urine. Tubular secretion is when the renal tubule extracts chemicals from the capillary blood and then secretes them into the tubular fluid. The purposes include acid-base balance, waste removal, and then the clearance of drugs and other contaminants. So the primary function of the nephron loop, also known as the loop of Henle, is to generate a salinity gradient that enables the collecting duct to concentrate the urine and then conserve water, meaning we're going to send water back to the blood. So electrolyte reabsorption from the filtrate goes as follows. The thick segment reabsorbs about 25% of sodium, potassium, and chloride in the filtrate. Those ions leave the cells by active transport and diffusion. Sodium chloride remains in the tissue fluid of the renal medulla, and the water can't follow because this thick segment is impermeable to it. So we're building up a lot of sodium in the medulla. The tubular fluid then is very dilute then as it enters the distal convoluted tubule. It contains about 20% of the water and 7% of the salts from the glomerular filtrate. If this was all passed by the time we got to the distal convoluted tubule, it would be about 36 liters of water a day. Again, that's still too much. So we've got to get rid of most of this 20% of water yet. We got to get that back into the blood. So the distal convoluted tubule and the collecting duct reabsorb variable amounts of water and salt and are regulated by several hormones. So this is kind of the area where we've got this 20% of water here to work with, and it's going to help us with how much we're going to regulate our water. Do we need more in the blood, or do we need to excrete more? And this is a good place to make that decision. So that's why this area is regulated by some different things like hormones, aldosterone, ADH, parathyroid hormone, and atrial natriuretic peptide. There's two kinds of cells in the DCT and the collecting duct. Principal cells, which are the most numerous and actually have receptors for those hormones, and they're involved in the salt and water balance. And then there's intercalated cells that are involved in the acid-base balance by secreting hydrogen into the tubule and the lumen and then reabsorbing potassium in exchange. Aldosterone is the salt-retaining hormone. Remember, this steroid is secreted by the adrenal cortex. And the triggers for aldosterone secretion are when your blood sodium concentration falls, or when potassium is too high, or there's a drop in blood pressure. Then there's renin that's released, which then becomes angiotensin II, and then it stimulates the adrenal cortex to secrete aldosterone. So the functions of aldosterone are that it acts on the thick segment of the nephron loop, the distal convoluted tubule, and the cortical portion of the collecting duct. It stimulates reabsorption of sodium and the secretion of potassium. And water and chloride follow the sodium. So there's that same idea again, that if we put sodium into the blood, water will follow it. Chloride will also follow the sodium as well, again, because remember, the negative is attracted to the positive. So the net effect is your body retains more sodium chloride and then therefore more water put back to the blood, which will help maintain blood pressure and blood volume. The urine volume will be reduced and will have a little more potassium in it. The natriuretic peptides are secreted by the atrial myocardium of the heart in response to high blood pressure. And then 4 actions result in the excretion of more salt and water into the urine, and then reduces blood volume, and then therefore blood pressure. It dilates the afferent arteriole, which constricts the efferent arteriole and increases the GFR. It inhibits renin and aldosterone secretion. It inhibits the secretion of ADH and inhibits sodium chloride reabsorption by the collecting duct. So if we keep more sodium in the nephron, then more water will leave with the urine, and then blood volume should go down, and then blood pressure should go down. Antidiuretic hormone, remember that's secreted by the posterior pituitary. If you are dehydrated, or there's a loss of blood volume, and blood volume osmolarity rises, it stimulates the arterial baroreceptors and the hypothalamic osmoreceptors. This triggers the release of ADH from the posterior pituitary. ADH makes the collecting duct more permeable to water. Then water in the tubular fluid re-enters the tissue fluid and then the bloodstream rather than being lost in urine. So if we put, again, more water into the blood pressure should increase. That's why it's called antidiuretic hormone. A diuretic is something that you take that makes you lose more water. Antidiuretic means you retain more water. Parathyroid hormone, or PTH, is secreted from the parathyroid glands in response to calcium deficiency or hypocalcemia. It acts on the PCT to increase phosphate excretion, and that acts on the thick segment of the ascending limb of the nephron loop, and then on the DCT to increase calcium reabsorption. It increases the phosphate content and lowers the calcium content in urine. So when you lose more phosphate in urine, calcium ions stay in circulation instead of joining up with those phosphates to create bone tissue. And PTH also stimulates calcitriol synthesis by those epithelial cells of the proximal convoluted tubule. So in summary, the proximal convoluted tubule reabsorbs 65% of the glomerular filtrate and returns it to the peritubular capillaries. And much of the reabsorption is by osmosis and co-transport mechanisms that are linked to active transport of sodium. Sodium is the key. The nephron loop reabsorbs another 25% of the filtrate. The DCT reabsorbs sodium, chloride, and water under hormonal control, especially aldosterone and ANP. The tubules also extract drugs, wastes, and some solutes from the blood and secrete them into the tubular fluid. And the DCT completes the process of making urine, and the collecting duct conserves water as well. In this section, we're going to look at urine formation and water conservation. Our objectives are to explain how the collecting duct and antidiuretic hormone regulate the volume and concentration of urine, and explain how the kidneys maintain an osmotic gradient in the renal medulla that enables the collecting duct to function. The kidney eliminates metabolic wastes from the body but prevents excessive water loss. That's their job. As the kidney returns water to the tissue fluid in the bloodstream, the fluid remaining in the renal tubules pass as urine and then become more concentrated, much like the color of the font I've chosen here. So the collecting duct begins in the cortex, right here. It receives fluid from several nephrons. Then it runs through the medulla. Here it reabsorbs water back to the bloodstream, making urine up to 4 times more concentrated here. The medullary portion of the collecting duct is actually more permeable to water than sodium chloride. And then as urine passes through the increasingly salty medulla, water leaves by osmosis, which then concentrates the urine and conserves the water. How concentrated the urine is depends on your body's state of hydration. Have you had enough to drink that day, or are you a little bit on the dehydrated side? Water diuresis is the drinking of large volumes of water and can produce hypotonic urine or very diluted urine. The cortical portion of the collecting duct reabsorbs sodium chloride, but it's impermeable to water. And the salt is removed from the urine, but the water stays in. So then the urine concentration could be as low as 50 milliosmoles per liter. When you're dehydrated, you have hypertonic urine, or very concentrated urine, a high amount of solutes and wastes. It becomes more scanty, as they say here, more concentrated. And the high blood osmolarity, because you're dehydrated, you don't have as much fluid in the blood, it's hypertonic. It stimulates the posterior pituitary to release antidiuretic hormone. This then stimulates an increase in the synthesis of aquaporin channels by the renal tubule cells. So this means you're going to have more of those little water channels to bring water back into the blood to be able to conserve it, so then your urine is more concentrated. If your blood pressure is low in a dehydrated person, GFR will also be low. The ability of the collecting duct to concentrate urine depends on the osmotic gradient in the renal medulla. It may seem surprising that the extracellular fluid osmolarity is 4 times as great deep in the medulla as in the cortex. We would expect salt to diffuse toward the cortex until it was evenly distributed through the kidney. However, there's a mechanism that overrides this. The nephron loop acts as a countercurrent multiplier, which continually recaptures salt and then returns it to the deep medullary tissue. It's called a multiplier because it multiplies the osmolarity deep in the medulla, and a countercurrent mechanism because it's based on fluid flowing in the opposite directions in 2 adjacent tubules. Remember how the loop goes downward and then upward in an ascending limb? As fluid flows down the descending limb of the nephron loop, it passes through an environment in increasing osmolarity. Most of the descending limb is very permeable to water, but not to sodium chloride. Therefore, water passes by osmosis from the tubule into the extracellular fluid, which then leaves the sodium chloride behind in the tubule. The tubular contents increase in osmolarity, reaching about 1200 milliosmoles per liter by the time that fluid rounds the lowest bend of the nephron loop. Most or all of the ascending loop in its thick segment, by contrast, is impermeable to water, but has pumps that co-transport sodium, potassium, and chloride into the extracellular fluid. This keeps the osmolarity of the renal medulla high, and since water remains in the tubule, the tubular fluid becomes more and more dilute as it approaches the cortex and is only about 100 milliosmoles per liter at the top of the loop. The lower end of the collecting duct is actually permeable to urea, but neither thick segment of the loop or the distal convoluted tubule is permeable to urea. So urea is continually cycled from the collecting duct to the nephron loop and then back again. Urea remains concentrated in the collecting duct, and some of it always diffuses out in the medulla, adding to the osmolarity there, besides the sodium being there. All of that is visualized in this diagram here. The large volume of water reabsorbed by the collecting duct must be returned to the bloodstream. It's picked up and carried away by the vasa recta, but this poses a problem. Why don't the vasa recta always carry away the urea and the salt needed to maintain the high osmolarity of the medulla? The answer is that the vasa recta all form a countercurrent exchange system that prevents this from happening. Blood flows in opposite directions in adjacent parallel capillaries, and as it flows downward into the medulla, the vessels exchange water for salt, and water diffuses out of the capillaries, and salt diffuses in. But as the blood flows back up to the cortex, the opposite occurs. The vasa recta give up salt and absorb water. They absorb more water on the way out than they give up on the way in. So thus they don't subtract from the osmolarity of the medulla. In this section, we'll look at some urine and renal function tests. We should be able to describe the composition and properties of urine and carry out a few calculations to evaluate renal function. Urinalysis is the examination of the physical and chemical properties of urine and is often done in certain checkups or when checking for certain disorders. The appearance varies from clear to deep amber, depending on the person's state of hydration at the time of submitting the urine sample. The yellow color is due to the urochrome pigment from the breakdown of hemoglobin from the red blood cells. Cloudiness or blood could suggest a urinary tract infection, trauma, or stones, or could be contamination with something else. Pyuria is pus in the urine, and hematuria is blood in the urine due to urinary tract infection or other damage. Odor can also be checked. Sometimes bacteria will degrade urea to ammonia, so you'd be able to detect that aroma. Diabetics will have a sweeter smelling urine because of the glucose in their urine. You can also check specific gravity and compare it to distilled water. The density of urine ranges from 1.001 to 1.028 grams per milliliter. Osmolarity, remember the blood is 300 milliosmoles per liter, but in urine could be from 50 to 1200, depending on how hydrated the person is. pH should be 4.5 to 8.2. Usually it should be around 6 though, which is actually mildly acidic. It should have about 95% water and 5% solutes. It might be normal to find urea, sodium chloride, potassium chloride, creatinine, uric acid, phosphates, sulfates, some calcium, magnesium, even some bicarbonate, urochrome, and just a trace of bilirubin in urine, but it would be abnormal to find glucose, free hemoglobin, albumin, ketones, and bile pigments. For a volume, a normal individual, an adult, should have 1 to 2 liters a day. Polyuria is excess of 2 liters a day, where oliguria is less than 500 milliliters a day. That's less than half a liter. Anuria is 0 to 100 milliliters a day. Low output from kidney disease, dehydration, circulatory shock, or prostate enlargement could account for any of these low readings. At output of less than 400 milliliters a day, the body can't maintain a safe low concentration of waste in the plasma, which then leads to azotemia. So with diabetes, remember you're going to find glucose in the urine. Diuretics, that's any chemical that increases urine volume. Caffeine is a diuretic. It dilates the afferent arteriole, which increases GFR. Alcohol is a diuretic. It inhibits ADH secretion. Some other diuretics may act on the nephron loop and inhibit sodium, potassium, and chloride symport, which then impairs the countercurrent multiplier and reduces the osmotic gradient in the renal medulla. And the collecting duct is unable to reabsorb or put water back into the blood like usual. Diuretics, because of their ability to cause the kidney to release more water in the urine, can actually be used to treat hypertension. Sometimes it's one of the first things that is tried when someone has high blood pressure, is to give them a diuretic, which makes them release more water in their urine, which decreases the level of fluids in the blood and thereby decreases blood pressure. In this section, we'll look at urine storage and elimination. We should be able to describe the functional anatomy of the ureters, the urinary bladder, and the male and female urethra, to explain how the nervous system and the urethral sphincters control the voiding of urine, which is actually a lot like what we learned in the digestive system with defecation. Urine is produced continually, but obviously doesn't just drain out continually. We're able to store it. So urination is episodic, occurring when we are ready, when we allow it. The ureters are retroperitoneal. Those are the muscular tubes that extend from each kidney, collect the urine, and send it down to the urinary bladder. They're about 25 centimeters long. A flap of mucosa at the entrance acts as a valve and keeps urine from backing up into the ureter when the bladder contracts. The urinary bladder is a muscular sac located on the floor of the pelvic cavity. There's 3 layers. They're covered by parietal peritoneum superiorly and by fibrous adventitia elsewhere. The muscularis is 3 layers of smooth muscle, and the mucosa is transitional epithelium. This is important in the flexibility and the stretchiness of the urinary bladder. Rugae, like the stomach has rugae, those are the conspicuous wrinkles when the bladder is empty and it's not stretched anymore. It becomes more wrinkly. The trigon is the smooth surface triangular area on the bladder floor and is marked with the openings of the ureters and the urethra. A moderate fullness of a bladder is only 500 milliliters, but it could be up to 800 because of the material it's made out of. It can be highly distendable. It fills and expands superiorly. Here we can see the ureters, the detrusor muscle, the ureter openings. Here's the trigon. Here's the internal urethral sphincter, the urethra itself, and the external urethral sphincter, and then the urethral orifice in a female. Some people have been known to have kidney stones. It's also called a renal calculus. This is a hard granule of calcium phosphate or calcium oxalate, uric acid, or a magnesium salt called struvite. These form in the renal pelvis and can dislodge. Sometimes they're so small you don't even know that they've passed, but a larger one, you'll know it, can be very, very extremely painful when they're passing through the ureters. Some causes are hypercalcemia, meaning too much calcium in the blood, dehydration, pH imbalances, having frequent urinary tract infections, or an enlarged prostate gland causing urine retention. Sometimes, if it's not going to pass, people might take a stone dissolving drug. Surgery might be an option, or a non-surgical technique that can pulverize the stones with ultrasound called lithotripsy. The urethra is the tube that conveys urine out of the body. In a female, it's 3 to 4 centimeters long, bound to the anterior wall of the vagina. And then the external urethral orifice is between the vaginal orifice and the clitoris. The internal urethral sphincter is made up of the detrusor, which is smooth muscle, and it's involuntary. You do not have control over this one. But luckily there's the external urethral sphincter, where the urethra passes through the pelvic floor, and it's skeletal muscle, so you have voluntary control over this one. And if it's not a good time to go, then you don't. The male urethra is 18 centimeters long. It has 3 regions. The prostatic urethra, which passes through the prostate. The membranous urethra, which passes through the muscular floor of the pelvic cavity. And the spongy or penile urethra, which passes through the penis. Then again, there's the internal urethral sphincter, which is the detrusor or smooth muscle thickening, and then the external urethral sphincter, which is a skeletal muscle that you do have control over. A urinary tract infection, or a UTI, is called cystitis. That's an infection of the urinary bladder, common in females due to their short urethra. Bacteria are much more likely to enter the bladder then. Sometimes sexual intercourse can cause this. Then it can spread up to the ureter, which causes pyelitis, which then can go all the way to the renal pelvis. Pyelonephritis can reach the cortex and the nephrons and can result from blood-borne bacteria. Voiding urine is the act of eliminating it. Between the acts of urination, the bladder fills, and the detrusor, that smooth muscle there, relaxes, and the urethral sphincters are tightly closed. The sympathetic activity in the upper lumbar spinal cord stimulates the postganglionic fibers to the detrusor muscle to relax it, and the internal urethral sphincter to excite it. Then the somatic motor fibers from the upper sacral spinal cord travel through the pudendal nerve to supply the external sphincter to allow voluntary control. And micturition is the act of urinating, and the micturition reflex is the involuntary spinal reflex that partly controls the first 4 steps of urination. And here they all are. There's 8 total steps. So number 1, stretch receptors detect the filling of the bladder and then transmit the afferent signals to the spinal cord. The signals return to the bladder from the spinal cord segments S2 and S3 via the parasympathetic fibers in the pelvic nerve. 3, the efferent signals excite the detrusor muscle. 4, the efferent signals relax the internal urethral sphincter. The urine is involuntary voided if not inhibited by the brain. So then voluntary control takes over. For voluntary control, micturition center in the pons receives signals from the stretch receptors, and if it's timely to urinate, the pons return signals to the spinal inner neurons that excite the detrusor and relax the internal and the urethral sphincter, and then therefore the urine is voided. If it's untimely to urinate, signals from the cerebrum excite the spinal inner neurons that keep the external urethral sphincter contracted, and then the urine's retained in the bladder. If it's timely to urinate, the signals from the cerebrum inhibit the sacral neurons that keep external sphincters closed, and the external urethral sphincter relaxes, and then the urine is voided. By the way, you do not have to memorize all of that. There's times when the bladder is not full enough to go, but you can override that and go anyway using the Valsalva maneuver to compress the bladder. It excites the stretch receptors early to get the reflex started. Here we have a little ending story on renal insufficiency and hemodialysis. Renal insufficiency is a state in which the kidneys can't maintain homeostasis due to the extensive destruction of their nephrons. So things like maybe hypertension, hypoxia, poisoning, or prolonged ischemia, any of those things could cause renal insufficiency. Neurons can regenerate and restore kidney function after short-term injuries, but not always. But if they are, The other neurons that are working can hypertrophy to compensate for the lost kidney function. You can actually survive with just 1/3 of one kidney. That would actually be enough to be able to remove waste from the body. But when 75% of the neurons are lost, urine output of 30 milliliters per hour is not sufficient. Normal should be 50 to 60 milliliters per hour to be able to maintain homeostasis. This causes azotemia and acidosis, and uremia develops, and also anemia. So one of the things that could be done is hemodialysis for clearing out the waste from the blood. The waste will leave the bloodstream and enter the dialysis fluid as blood flows through a semi-permeable cellophane tube, and also removes excess body water in a machine that looks like this.